Let's talk about some other biotechnology techniques. Specifically, we're going to look at PCR and gel electrophoresis. Now, these are specifically mentioned in the syllabus that you need to be able to explain the purpose of these processes. So while I'm going to go through and explain how each of these processes do work, you're not required to actually remember the process, more about, well, why would we want to do it? Our PCR, super well known at the moment for COVID tests. And PCR effectively allows us to create millions of copies of DNA from a single DNA segment. You can see this picture overview here. If we've got a DNA sample, some primers, some nucleotides to make the new DNA from, a buffer and some polymerase, which you guys all know about, then we can effectively take that DNA sample and multiply it many times. How does this work? That's what we're going to look at. We're effectively doing what your own body does in terms of DNA replication, but amplifying it by a significant amount. And we're using it to copy a DNA segment that we want. So it makes millions of copies of that DNA from a small sample. The example given here is that it could be DNA found at a crime scene. It could be a gene that a researcher is experimenting with and needs large copies of, such as we were talking about with recombinant DNA, because maybe you want to make some recombinant plasmids. But most commonly these days, as we would know, is it's used for PCR testing. By taking a swab from your throat and your neck, we are able, not neck, nose, my apologies, we're effectively able to take that sample and amplify it so many times that we're able to easily read it. And then from reading that DNA sample, we can see, well, do you have the COVID DNA inside of you? Process needs polymerase, of course, to make the new DNA, and that comes from bacteria that are really heat tolerant. So we have this Taq polymerase, which is a specific type that comes from thermophilic bacteria. And that means that the polymerase enzyme doesn't denature at high heat, because we do need high heat for PCR. As I said, it's basically like normal DNA replication. In normal DNA replication, we have primers. We didn't talk about that too much, but little starting elements. So the experimenter will pick a primer that is about 20 base pairs long that then attaches to our target DNA. From there, once the primers have bound to the target DNA, the Taq polymerase is able to then extend it and make a new section of DNA, copying it. Okay, so we attach the primers, and then the Taq polymerase comes in to make the new DNA, much like in normal DNA replication. And you can see that it's working only in the one direction, as we know that polymerase does. So we've got it copying in opposing directions for the two different strands. But we do need to denature that and split it up first, as mentioned. For it to happen, we need these ingredients, as I said earlier. A thermocycler basically heats it up, cools it down, different temperatures for different parts of the phase, and everything else is to help the process along. Either ingredients, the primers and the nucleotides and the polymerase, or the buffer just helps keep it all mixed together. The steps are first and foremost, denaturation. So we need to denature the DNA so that it separates. We don't have our helicase and topoisomerase that we would normally have in our cell. So we need to heat the sample so that the DNA separates. So we've got single-stranded DNA templates now. Then we have the annealing process, where they cool it down from that really high temperature, 96 degrees, down to between 55 to 65. And by cooling it, that allows the primers to then move over and attach to the DNA. Okay, so annealing, they're attaching there. And then the last stage is the extension phase. We heat it up to 72 degrees, which will allow the Taq polymerase to then copy the DNA and synthesize new strands. Okay, after one cycle, we go from one strand of that DNA to two, or if we had more than one strand in our original sample, which is more than likely the case. Let's say you take a COVID swab from your nose, you could have 50 to 100 COVID virus particles in your nose. So we're taking, 50 to 100 and duplicating it. So if we have 100, we're duplicating it to make 200 after that. That 200 is then duplicating again. And typically, we do this process for 25 to 35 minutes. The purpose for this is pretty straightforward. The purpose of this process, PCR, is just to make bulk amounts of DNA. So it's to make enough of your target DNA so that it can be analyzed or so that it can be used. So there's two essential purposes there, but at the end of it, it all boils down to making lots of DNA. We can use this to make the DNA product so that it can then be analyzed, such as for a crime scene analysis or for viral analyses, or we can copy it so that we have enough of our target DNA for processes such as gene transformation. But if we were looking at analyzing the DNA, we can then go through processes such as DNA sequencing or DNA profiling, which allows us to analyze what is actually in the DNA. We can clone it to make recombinant plasmid DNA, and we can also visualize what is there using gel electrophoresis. So that analysis using gel electrophoresis allows us to see what we actually have in our DNA. Has the result been successful? Have we produced what we want? On the PowerPoint, there is that link there to a virtual PCR lab. It takes like 5-10 minutes, but it gives you a really great understanding of it. I strongly suggest you all go and check it out. Gel electrophoresis is basically like chromatography. It is separating based on the size of our molecules. It's an analytical technique that we often use after PCR, or we can even use after DNA cloning. Once we've made a lot of our recombinant DNA through DNA cloning, or once we've copied all of our genes through PCR, then we can basically test to see, have we got what we want? And the way this is done is by loading it onto a gel there and by running an electric current through it. And to simplify, the larger molecules will not be carried as far with that electric current, whereas the smaller molecules will travel further because they're lighter. So the gels are placed into a gel box with a buffer solution that covers over the gel, and that allows for movement and for electric current to flow through. The samples then placed into those wells in the gel. You can see in the image there at the top, the rectangular areas are our wells. They're just holes within the gel that allow us to insert our samples. Electric current runs through it, and then the DNA will separate out based on fragment size. The smaller fragments move more quickly and go further. From there, we can work out what the DNA ladder of our sample, and we can work out the size of the bits of our DNA there, which allows us to analyze them. So the purpose of this is to be able to visually analyze the contents of our DNA sample. It's not necessarily going to show us these are the sequences in our DNA, or these are the genes present, but it will allow us to assess some physical aspects of our DNA sample. And if you're a geneticist or a crime scene analyst, something like that, then you have the tools to understand this. Primarily, it's used for checking of the products of PCR DNA cloning to see, has that process been effective. But it can also be done to create a DNA profile of someone, because when you compare the results of gel electrophoresis of the DNA of multiple people, then we can compare how closely related they are. If we look at the sample on the right there, just to give you a little bit of an opportunity to analyze this, okay, which sample contains the longest fragment, the shortest fragment, and then a fragment with 1,500 base pairs. So that's the length. So the longest fragment, which is 3,000 base pairs, is only found in sample 1. The shortest fragment, which is probably about 100 base pairs, is only found in sample 2. And if we have a look, sample 3 has a 1,500 base pair DNA fragment. Again, there is a fantastic virtual lab on this that I strongly suggest you take the time to have a play around with. It won't take long out of your life, and it will make this seem a lot more real and grounded for you. Our next two things to look at, our next two and our final two biotechnology techniques are DNA sequencing and DNA profiling. And all the criteria ask you to be able to do, the objectives ask you to be able to do for this is basically recall examples of when this is used. DNA sequencing is the most involved of DNA profiling, DNA sequencing. It basically takes the exact nucleotide sequence of an entire organism or person's DNA. So it allows us to get an exact mapping or sequencing of their entire DNA. That allows us to then compare them to other people. We can look for any substitutions, any deletions, any additions, mutations, basically, if we're looking for, well, why does this person have a particular trait that they don't normally have? And we can also use this to track common genes between people and organisms. This is used often for determining evolutionary relatedness. So there's algorithms. Once you've sequenced the DNA, you can put it into a computer which automatically assesses the similarity. And if we can look at the amount of base pairs different between different organisms to see how closely related they are. And this is the foundation of modern cladistics and how phylogenetic trees or cladograms are produced in the common day. And a lot of our old evolutionary trees are being rewritten thanks to genetic evidence as we understand how closely related they are. We can also use it in medicine for looking at the genes that cause diseases. So you can use it for genetic testing to see if you're prone to a disease. It's used often in agriculture, mostly to look at how pure or what particular strand certain crops are. It can be used in paternity testing, for breeding, and forensic ID as well. If we look at a particular focus here, and that is medical diagnosis, we can use DNA sequencing for that medical diagnosis, as I mentioned, with COVID-19. So we're able to use PCR to amplify a sample taken from someone and then make multiple copies of that DNA. Then we can look at the results through DNA sequencing to figure out, well, does this have the particular sequences we know that COVID's DNA has? So we are able to PCR and then DNA sequence test to find out if this person has RNA. And this process is the RNA from the virus. This process has been getting better and better as time has gone on with COVID. As well as this, it can be used for PGD, pre-gestational diagnosis, which is basically looking at how likely is it for an offspring to have certain genetic diseases. It can be used to look at what type of cancer a person has. It can be used to look at their susceptibility to certain diseases, such as the BRCA1 gene, which predisposes you to breast cancer. We can also use it in this incredibly awesome new field of eDNA, or metagenomics as it's also called, environmental DNA, and it takes samples of DNA from the environment. A friend of mine who's a marine biologist in Saudi Arabia was out on the Red Sea recently, and they would take water samples while they're out there. They would take their water sample and they would run it through probably a PCR machine so that they've got lots of DNA there to analyze, and they would run DNA sequencing. And that DNA sequencing allows them to look for sequences in that DNA to help identify what animals are there. Sure, you might not see a killer whale, but if there's been a killer whale around the area, it will have left behind some of its DNA. And we can identify that killer whales are in that area. So it can allow us to identify what organisms might have lived there. It can allow us to identify what organisms currently live there, and a whole bunch of other fields. But that's just one of the many fields that DNA sequencing is incredibly useful for. And this one is being used more and more in the common day. DNA profiling is a little easier. We're not looking at the entire DNA sequence. We look for specific sections of the DNA. So we're comparing natural variations within people's genomes, and we can use it most often to match DNA samples found at a crime scene to suspects. So what they do is they look for STRs, short tandem repeats, which are non-coding sections of the DNA that show high variability between people. So that means that they're likely to virtually always be different between different people. If we take a DNA swab from a crime scene, we compare it to somebody who left that DNA at the crime scene, then it will be identical. If we look at it, say, somebody who's closely related to them, then it will show high similarity with some variability. And the more distantly related these people are, the greater variability. So we can use this for maternity and paternity tests, but it's also often used for forensic ID. And these are the other applications of it. So forensic ID, paternity, maternity, or other kinship identification. This is the sort of thing that they'd often be doing in those 23andMe or Ancestry tests. We can look at genetic pedigree of animals in terms of someone selling horses that they claim came from a particular stud, and we can test if that's true. We can look at how organisms are mating and reproducing by taking the genetic samples and then profiling them from multiple individuals within a population. We can see how are they breeding. So are they breeding relatively similarly, or is one male basically dominating the breeding time with all the females? We can also look at tracking things like pollination, spread of plants, etc., various other uses of this. That's it in terms of our the other biotechnology techniques we want to go through. The only other objective for this unit is one that's best done in class, and that's appraising data from an outcome of a current biotechnology technique to determine its success rate, effectively analyzing data, much like you would be doing in your IA3s, which is coming up. Okay, so from this unit, you should be able to describe the process of making recombinant DNA in those 4 key steps. You should be able to explain the purpose of PCR and gel electrophoresis. Why would a scientist want to undergo these processes? And then recognizing the applications of DNA sequencing to map species genomes, and then DNA profiling to identify unique genetic information. All right, that is it for this unit and for topic 1 of unit 4.